welcome to this video. Today I'm going to talk about genome-wide methylomic regulation of multiscale gene networks in Alzheimer's disease. DNA methylation is one of the major epigenetic processes to regulate chromatin structure that controls transcriptional activation or suppression of genes. In this study, we try to address the following research questions. First, what is the overall landscape of DNA methylation in Alzheimer's disease? How does DNA methylation impact individual genes and proteins in Alzheimer's disease? Third, and how does the DNA methylation impact high-order interaction gene and protein networks in Alzheimer's disease? We previously established a Mount Sinai cohort of large-scale genomic, transcriptomic, and proteomic data in Alzheimer's disease. So in this study, we extended this cohort by generating DNA methylation data to provide 850K CpG probes from about 200 samples in the perhippocampal gyrus of control and AD brains. First, we compare AD and control samples to identify about 14,000 and differentially methylated CpG probes. And then we identify 270 differentially methylated regions, which are comprised of highly correlated adjacent CpG probes. To summarize, the overall impact of 270 DNA methylation regions on each gene or protein, we summarize the correlations between a given gene or protein with all the 270 DNA methylation regions. We separate positive correlations from negative correlations, which correspond to hypo and hyper methylation. Therefore, we got an overall hypo methylation score or hyper methylation score for each gene and protein. We further calculate the overall impact of 270 DNA methylation regions on the gene or protein modules from the network analysis by summing up the overall hypo or hyper methylation scores from the member genes of each gene or protein module. We previously established the global networks and the potential drivers for Alzheimer's disease using the RNA-seq data from the same Mount Sinai brain bank and Alzheimer's disease cohort. So now we identify hundreds of genes, modules, and identify the drivers. So get back to DNA methylation, we found out that the overall module methylation scores are highly correlated with the module relevance to Alzheimer's disease. As we separate hypomethylation scores from the hypermethylation scores. So here we basically see two sides of modules. One side of module is downregulated in Alzheimer's disease. They have very high hypermethylation scores, while the upregulated gene modules, they have very high hypomethylation scores. So this figure basically shows the differential expression and also the DNA methylation. It is a little bit complicated, but the key message is that there is a good consistency between the differential expression and the differential methylation. We first look at the relationship between the key regulators from the network analysis and the driver regulators, overall hypo or hypo DNA methylation scores. Again, we see very good correlations between the key drivers' relevance to Alzheimer's disease and the key drivers' DNA methylation scores. So the take-home message is that DNA methylation is a key regulator of molecular changes in Alzheimer's disease, and they are the key regulator for the interacting gene and protein networks. Yeah, I would like to thank the team members from Mount Sinai, St. Jude Children's Research Hospital, Columbia University, Rush University, and also the funding institute, NIH, NIA, as well as the AMP-AD consortium. Especially, I would like to thank the two co-first authors, Dr. Ermin Wang and Minhui Wang, for their wonderful work in this study. Thank you.